this is Miss Dagenford, and we're going to go through our cell cycle and DNA introduction, and that our goal for this video would be to describe why cells stay small, to identify the parts of the cell cycle, and to describe the parts of the cells that are involved in cell division. Some of these you already know before, and others we're going to get into and just develop the idea a little bit more. Now, the first thing we want to tackle is why cells need to divide in the first place. And it has a lot to do with surface area and volume. Now, these are terms that you've probably talked about in your math classes. Surface area would be the area that is the surface. That would be representing the cell membrane. The cell membrane envelops the outer part of the cell. It would be similar to wrapping paper. If you've got a present to wrap, the wrapping paper would represent the surface area. And as the cell gets bigger, the surface area gets bigger as well. Much like having a bigger present requires more wrapping paper. Now, volume is another measurement that we want to look at as well. Now, the volume would be the inner part of the cell. Different from the surface area is only the wrapping, the volume is all the inside. So just like the inside of a stadium, you have a lot of people crammed on the inside. And so imagine a stadium full of people, well, that would be the volume. But the outer layer of the stadium, the wall of the stadium, would be the surface area. Now, as a cell gets bigger, and if you think about it, this is an interesting question. Why aren't we just a great big giant one-celled organism, a 5-foot cell? Like, why aren't we one cell? We start out as a cell, right? A sperm cell unites with an egg cell, good old middle school, right? Sperm cell fertilizes an egg cell, yay, one cell zygote. Why don't we just get bigger and bigger and bigger? And it has to do with our ability to fuse nutrients and oxygen and then get rid of carbon dioxide. The bigger the cell, the more volume there is to try to take care of inside the cell. And so by keeping the cell small, we get more surface area per volume. And that keeps the cell more efficient. So the smaller the cell, the more efficiently we can deliver oxygen, the more efficiently we can deliver food, and the more efficiently we can get rid of carbon dioxide in the case of animal cells. And so much like a stadium, if you have a really big stadium, you've got to make sure you've got plenty of doors in the stadium walls to let people out. And so the bigger the stadium, the more problematic that is. If we had a really small room, it doesn't make that much difference. So the smaller a cell is, the more efficient it is. Now, so now we know why cells divide. They can't get too big; it becomes more of a problem. So, what are the things involved in cell division? What are the cell parts involved? So let me just keep going here. So one of the things that we need to concern ourselves with cell division would be our chromosomes. Chromosomes carry our DNA. And so you can imagine it becomes hugely important to make sure our DNA gets distributed properly. You shouldn't want a cell to divide where one cell gets extra DNA and the other cell is missing DNA. That would be horrible. So the most important thing in cell division is to make sure that the cells get all of the right chromosomes. So let's go over some of the terminology that we're going to be using with our chromosomes. A chromosome would contain genes, and the genes are going to be important for making cell parts. And what they look like right now isn't very impressive. They just look like a line. They're linear. And so in the red up here, we've got one chromosome. And what ends up happening, in order to divide, the cell has to make a duplicate copy of each chromosome. So we go from having just one copy in the red to two copies. And you can see they changed the color a little bit, dark red to a pink. Now, down here in the blue, we see the same thing. We've got one chromosome, and then we have a little area in the center called the centromere. When it comes time to divide, the cell needs to copy the chromosome. And so here we go, we're going to make a copy. And they're tethered in the middle of in the centromere. The only tricky thing is how we call these things. The one chromosome over here on the left, we simply call one chromosome. I know, that's not very hard. However, when we duplicate this chromosome, we still refer to it as just one chromosome. That's probably the weirdest thing about this slide. Over here on the left, that's one chromosome. We make a copy, they are tied together like that. That's still one chromosome. Now, we call each copy a sister chromatid, but it's one chromosome. As soon as they divide, then they're considered two chromosomes. You're going to get a lot of practice from that. So, the one on the left here, one chromosome. The one on the right, still one chromosome. The one up top in the red, one chromosome. The one right next to it, one chromosome with two sister chromatids. As soon as they divide, two chromosomes. I know, we'll practice that. Now, some of these genes control cell division, and you will have either watched the videos on cancer or are going to watch the video on cancer, and you're going to see that if the cell can't control the cell division, we've got a problem because the cells divide uncontrollably. Now, the purpose of cell division is to split the chromosomes and properly distribute them. Each cell has two sets of chromosomes, one from mom, one from dad, and we call that set diploid. So if you have two sets, you are a diploid cell. And you can count them up. Humans have a diploid number of 46. So you'll notice 2, 4, 6, 8, 10, 12, blah, 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 to the last two, 46. And so the purpose of cell division is to copy each one of these, and each daughter cell will get 46 chromosomes. Now, we call the whole start to finish the cell cycle, and cells go through these stages of growing and then dividing. And they divide when they get a certain size and certain signals. There are checkpoints to make sure that the cell is not going to duplicate when there's damage. If there's damage, the cell needs to stop and either repair itself or it will self-destruct. And so we're going to see some of these checkpoints. Now, cells undergo cell cycle at different rates. Your skin cells, your digestive cells, your blood cells, or the cells that make the blood cells, they divide every few days. They divide very rapidly. Every few days, you recycle your skin. But your nerve cells and your muscle cells, they don't divide at all. So different cells go through different rates of cell cycle. So let's go over these. The first one, and you'll notice on yours, you'll move the slide. So make sure you move the slide so you've got G1 labeled. G1 would be growth 1, where the cell is carrying out normal cell functions. The DNA is nice and open and being used. The cell increases in size. And by the end of G1, there needs to be some checkpoints. Are there enough nutrients? Is the DNA undamaged? If everything's good, the cell goes to S phase, where the DNA replicates. And at that point, we get the single chromosomes that now look like X's. They are duplicated chromosomes. It would still be 46 chromosomes, but duplicated. Now, the checkpoint here is that the DNA replication happened correctly. If it did not, a proper cell would stop at this point, either fix it or self-destruct. We cannot have cells dividing with damaged DNA. That's not good. All right, if everything's good, we're going to go through the second growth phase, G2. And again, move your slide so it's labeled G2. The cells are prepping for division at this point, as long as everything's still good. Normal functioning is still happening. If it's a skin cell, it's protecting. If it's a muscle cell, it's contracting. If it's a digestive cell, it's secreting acid. So normal functioning is still happening, but it's also starting to prepare for cell division. The checkpoints are still there. All organelles better be duplicated. The DNA is still undamaged. If everything is good, we're going to go through mitosis. And mitosis is technically where the nucleus divides. The nucleus arranges the chromosomes and then divides the chromosomes to each of the daughter cells. At this point, you still have checkpoints to make sure that everything is okay. Everything is arranged correctly. You can still stop the cell at this point, and if everything's okay, it'll go on. If something's wrong, it needs to stop. Now, mitosis is then followed by cytokinesis, where the actual cytoplasm divides. The organelles get distributed, and the cell membrane divides the cell. And we'll look at each one of these. Now, G1S and G2 are also called interphase. Interphase would go through the whole three of these. And the interphase was the original name back in like the 1850s, 1930s. We didn't know all of these things were going on. We just knew that the cell wasn't dividing. And so we call it interphase. Then let's see what the end result is. We start with a diploid cell. In humans, that would be 46 chromosomes. So we start with a diploid cell. We end with two daughter cells that are each diploid. And so they should have the exact same copy of chromosomes. Why do we divide the cells? Well, not only because we've gotten too big, but why does our body have cells divide? Well, we repair cells. If we cut ourselves, we're going to repair cells and heal. We're going to replace cells. Our skin cells are dividing. And then we're going to grow and develop, and mitosis is doing that. Mitosis makes basically your entire body cells. It replaces cells, it grows cells. Now, the silly way to remember this is mitosis makes mitose. And that's an easy way to remember that mitosis is just the cell division that makes an exact copy of the cell. So, I hope that gives you a nice introduction to our cell division.